Hello, and welcome to our Bio46 Lichen Chronicles by Emily and Layo, where we'll briefly walk you through some studies centered around some seemingly unlikely agents in the fight against climate change and our journeys through forests and shrublands to find them. Disclaimer, I apologize in advance for realizing too late that most of the videos we took on our trips were recorded vertically and spontaneously, but it's just to get a feel for the surrounding areas and climate and how that factors into our studies. So, as we should all know by now, air pollution is one of the most significant markers of the human impact on the environment. An increasing body of scholarship has begun to scrutinize the less apparent ramifications of air pollution in our world, but relatively few studies have explored the effects of pollution on overall ecosystem health and biodiversity, especially through the lens of ecological community interactions. Our work focused specifically on parasitic interactions, where one organism benefits at the expense of another, and the effect of air pollution on these interactions is an unsettled question. So, enter the previously mentioned agent in question, lichen. What are they and why do they matter? To put it simply, they are symbiotes made of algae and fungi living together mutually. Due to their dependence on the environment and the atmosphere as a primary source of nutrients, lichens are highly sensitive to air pollution and make really good candidates in climate change studies as a result. Some species vary in sensitivity, which allows an additional layer of determining pollution levels by seeing which are present in a certain area. But on top of that, lichen can be infected by external parasitic fungi. Out of the various factors that can disrupt the lichen health, little is known about whether they relate to one another and if infection could indicate anything at all about pollution levels, and that became the main question of our experiment. We hypothesized that high pollution levels would correlate with high rates of the lichen species Ivernia prunastri that are infected by the fungus Unguiculariopsis latae, which we will just refer to as fungal infection or external fungus to make things easier. Furthermore, we thought that this connection will be caused by air pollution adversely affecting the microbiome diversity and photosynthetic capacity of the host lichen, reducing immune response of Ivernia to external infection. And to find answers, we sampled lichen from multiple trips to 3 preserves, Jasper Ridge, Arastradero Preserve, and the Stanford main campus area. We also collected environmental data at each sampling site using sensors recording temperature and humidity trends over the course of 2 months. All sampling was conducted during the January and February months in the winter season, when precipitation levels and lichen growth and activity rates are usually at their highest. We also compiled data from past Bio46 classes from 2019 and 2020 to more clearly analyze trends. And on these trips, we noticed very evident environmental differences at the sites that we were assigned, which also added to our speculation to what affected lichen health. So our first Jasper Ridge trip was a damp, shady forest with a nearby river and even some redwoods nearby. The lichen were noticeably more green and somewhat damp as well, and fungal infection levels varied. Overall, it was just very moist and lush. And on the other hand, our Arastradero site was very elevated, exposed to the sun, hot and dry. I do regret deleting a video I took from the hike to get up there, but it was a very stark contrast to Jasper Ridge. The lichen were confined to this mostly dead oak tree, and although they were dry or not healthy looking, there was not too much infection. It was also hard to find specifically Ivernia, implying that there were higher pollution levels from being closer to busy roads. Our collection site of the second Jasper Ridge trip was also elevated, exposed to the sun and dry, similar to our Arastradero site, but there was more dense shrubland where there were far more Ivernia there that appeared to be in moderate health. Finally, our site near the Stanford Dish, which was a tree grove at a very interesting incline, was by a small road that we biked up to get there but had a decent amount of Ivernia present, showing that maybe there wasn't enough car traffic from the small road yet to produce a pronounced pollution effect. But regardless, there was only so much that we could speculate from observations, so we plated the samples and were sent to the labs to get tested for a myriad of variables, and out of the ones that were applicable to our hypothesis, we obtained the following data and results.
Our results, however, presented a less clear picture. In short, we tested 4 hypotheses, and the results were 1. Increased air pollution is slightly correlated with decreased proportions of infected lichen samples, though this relationship is not statistically significant. 2. Increased air pollutant levels negatively impact the photosynthetic capacity of lichen tissue. 3. Increased levels of air pollution are weakly correlated to decreased levels of taxa richness in lichen, but this relationship is not statistically significant. And 4, decreased lichen microbiome diversity and photosynthetic capacity are both correlated with increased fungal infection rates. Overall, we found that there was not a strong correlation between air pollution and fungal infection rates. We thought this might be due to the fact that air pollutants can have both a positive and negative effect on photosynthesizing organisms depending on their concentration and the type of substance. There was, however, a clear effect of task diversity and photosynthetic capacity on fungal infection rates, suggesting that the two variables do play a part in the lichen's immune response process. These findings present promising mechanisms that may explain how Avernia maintain a strong immune response to necrotrophic and fungal infection, as well as how different forms of air pollution may bolster or inhibit these responses.
Overall, the uncertainty is a good reason as to why further research on these seemingly unlikely environmental indicators will always be welcomed, and while we didn't get a direct answer about fungal infection serving as another indication of polluted lichen, we still gained a variety of intricate knowledge about these organisms and their relationship with the environment.